Good afternoon. I'm Gallego Cortés, and today I will be presenting the project whose title is Functional Myeloid-Derived Suppressor Cells are maintained during antiretroviral treatment and preclude HIV-1 reservoir reactivation. Antiretroviral therapy effectively suppresses HIV replication in people living with HIV, providing a control over the infection. However, this antiretroviral therapy fails in eliminating HIV from the body due to the persistence of HIV-latent infected cells. In the past years, several therapeutic strategies aimed at depleting the latent HIV infection have gained more attention. One of these interventions involves the pharmacological activation of dormant viruses, leading to killing of infected cells by viral cytopathogenic effects or by the action of the immune system. This novel strategy, known as shock and kill, uses latency reversal agents as a means to reactivate the latent HIV. In fact, these latency reversal agents have been proven to be partially successful in reactivating the latent HIV in vitro using PBMCs from people living with HIV and in vivo during clinical trials. In addition, immune effector cells can selectively identify and eliminate latent reactivated CD4 positive T cells that have been latently infected in an in vitro setting. However, so far, all the arrays that have been tested during clinical trials have failed in reducing the HIV latent reservoir. The inability of these latency reversal agents in eliminating these HIV-infected cells may be due to the presence of in situ immunoregulatory mechanisms that inhibit viral reactivation and the action of anti-HIV immune effector cells. In this context, multiple cells can contribute to immune suppression during viral infections, such as regulatory T cells, NK cells, and myeloid-derived suppressor cells. In the past years, myeloid-derived suppressor cells have gained more attention due to their association with progression of chronic infections such as HIV. These myeloid-derived suppressor cells are a heterogeneous population of immature myeloid cells that originate under pathological conditions such as cancer, autoimmune disorders, or autoinflammatory diseases. They have a key role in preventing excessive immune activation by modulating cell types such as NK cells, T cells, dendritic cells, and B cells, and they exert these immunosuppressive activities through several mechanisms, such as the production of arginase and indoleamine 2,3 deoxygenase, or IDO. Characterization of these cells in the context of HIV infection remains limited. However, it is plausible that persistent HIV infection induced the recruitment and generation of MDSCs in a similar manner to the one observed in the context of tumors. Indeed, there is evidence that MDSCs are implicated in HIV infection progression. It has been observed an increased frequency of MDSCs in people living with HIV, positive correlations between regulatory T cells and viral load and these cells, and increased arginase activity in these MDSCs, and also an increased IDO activity in both viremic and AR-treated patients. Based on these findings, we propose the following hypothesis. MDSCs promoted immune tolerance against latently infected CD4 T cells may play a critical role in preventing viral reactivation and clearance. To address this hypothesis, we propose the following aims: to phenotypically characterize different populations of MDSCs from both healthy donors and people living with HIV, both viremic and on antiretroviral treatment, and to examine MDSC's capacity to inhibit CD4 T cell activation and viral reactivation in latently infected CD4 T cells ex vivo. First, we perform the phenotypic characterization of different subsets of MDSCs from the three cohorts that I previously mentioned: healthy donors, people living with HIV, both viremic and on antiretroviral treatment. And this is the gating strategy we use to identify these subsets. We first gated the CD3 negative cells that were positive in CD33 and CD11b, and that exhibited low or negative expression of HLA-DR. Then we distinguish between monocyte-like myeloid-derived suppressor cells and granulocytic-like myeloid-derived suppressor cells based on the expression of the CD14, being the monocyte subset CD14 positive and the granulocytic subset CD14 negative. Our first analysis showed that there were no significant differences in the frequencies of monocytic myeloid-derived suppressor cells among the three groups. However, we did observe a significant reduction of these cells in the viremic group. Then we examined the immunosuppressive capacity of these two groups of myeloid-derived suppressor cells based on the expression of arginase and IDO. We reported an increased arginase expression in both subsets of myeloid-derived suppressor cells from both viremic and untreated patients, but only an increased IDO expression in the granulocytic subset and in VHIR patients, in viremic patients. We also saw a positive correlation between the monocytic myeloid-derived suppressor cells expressing arginase and the regulatory T cells, and positive correlations between the arginase expression in the granulocytic subset and HIV DNA and RNA, which are disease progression markers. Then we performed functional assays with these two groups of MDSCs. And in these assays, what we did is isolate CD4 positive T cells from people living with HIV on antiretroviral treatment and reactivate these CD4 T cells using PMA and ionomycin for about 20 hours. The following day, we isolated the two subsets of MDSCs and co-cultured them with the reactivated CD4 T cells in a 1-to-1 ratio for about 20 hours. On the final day, we assessed viral reactivation and CD4 T cell activation based on the expression of the viral protein P24 and the activation marker CD69. Here I provide a representative example of the gating strategy that we used to study both the CD4 T cell activation and the HIV reactivation. And from these assays, we observed that both subsets of MDSCs were able to reduce CD4 T cell activation, but only the GMDSC subset was able to significantly reduce the viral reactivation. In addition, when we added an arginase inhibitor to the co-culture of the GMDSCs and CD4 T cells, we observed that viral reactivation seemed to be restored. However, we need to perform more experiments in order to corroborate this observation. To date, our conclusions from this project are that MDSCs from people living with HIV exhibit higher immunosuppressive activity compared with healthy donors, that HIV infection significantly expands the population of monocytic-like myeloid-derived suppressor cells expressing arginase and granulocytic-like myeloid-derived suppressor cells expressing both arginase and IDO, and these populations are not normalized after the antiretroviral treatment, that there is a positive correlation between the monocytic subset expressing arginase and regulatory T cells, and a positive correlation between the granulocytic subset expressing arginase and HIV DNA and RNA. We also observed that both populations of MDSCs could significantly decrease the CD4 T cell activation, but only the granulocytic subset could inhibit viral reactivation. And it seems that the inhibition of the arginase activity in this last subpopulation seemed to restore HIV reactivation. Overall, we concluded that persistent HIV infection is associated with an expanded population of GMDSCs arginase positive, which impedes viral reactivation. And we think that finding new therapeutic strategies that are able to modulate the immunosuppressive action of these cells can have an effect on the HIV reservoir of these patients. Lastly, I have to thank everybody on my group, the study participants, and also all the funding bodies, and for you to listen to my presentation. Thank you.
Thank you very much. Is there any questions? No? Okay, I have one. I really like the functional assay, but I was wondering, I mean, you're using a 1-to-1 ratio, so I was wondering if that's like the proportion of the cells in the natural setting, or if you're increasing a little bit the amount of...
I am increasing the amount of cells, yes.
And have you done like a lower proportional and more physiological maybe ratio to check those findings?
I did test different ratios, but because it's really difficult to see HIV reactivation in people living with HIV that are on antiretroviral treatment, we need a lot of cells to see if there is an inhibition. And also, even though I said it's a one-to-one ratio, there are not that many cells in these patients, so they are kind of tricky, these assays.
Yeah, you had like a lot of vials.
Yes.
Depicted, not for the CD4s, but for the suppressor cells, so I guess it's hard.
Mm-hmm.
Okay. Are there any other questions? No? Okay.
Yes, the inhibitor of arginase is for both isotypes of arginase. Arginase is for 1 and B, and which is the one that is expressed in the cell type?
Yes, it's the inhibitor for arginase 1 that it has been described that are in this enzyme, in this cell type.
And the two isoforms, they have overlapping roles, so they have completely different roles.
I really don't know actually, but because we are only studying this one, but I will check it. Yeah, thank you.
Thank you.
Any other questions? No. Okay, just a final question. So you were mentioning that by using particular compounds that would target these cells, you might be able to enhance the reactivation. So do you have any clue about which kind of compounds could be working?
Well, I wanted to suggest that arginase inhibitors, but I don't think that they will be selective for these cells. So I guess as these cells, they don't only have one mechanism in inhibiting these T cells or dendritic cells or whatever. I guess we should attack many different mechanisms. However, they have to be pretty selective because the problem with T cells is that they share many markers with several other cell types. So it's kind of tricky to attack these cells, yeah.
Okay, well, good luck then.
Thank you.
Thank you very much.